So ChIP-seq downstream analysis, after, the reason people are doing ChIP-seq is they want to know what is this transcription factor binding to, and what does it do, right? So there are a number of these interesting things you want to do. The first is you want to look for a motif. In fact, if you have ChIP-seq motif analysis, it's much easier. It's also a very important QC. You can run meme, you can run other method. Yeah, so we have our own motif finding algorithm, which is used to find motif in all the public ChIP-seq data that we collected. In addition to know that the motif should be enriched in your data compared to the genome background, what we also know is that this motif, if you look at your peak, fold change in terms of enrichment, this motif should appear more often in the peaks that have higher fold change than lower fold change. It kind of makes sense. If that region has 10 copies of the motif, you do ChIP-seq, you'll have multiple transcription factor binding in that location, then you have a higher fold change. In addition, if your peak, you remember your peak is like this, very often the motif should appear more likely to be in the middle compared to the side, because the side is just some random garbage, right? The transcription factor is usually binding right at the peak. And so we do use these two considerations to help us decide which motif is really good. And that tool is already in Systrom. And one thing we notice that the transcription factor in the same family often have similar binding motifs. And but you can also use the ChIP-seq motif to find transcription factor interactions. For example, this is the example for the same TF in the same family have the similar motif. You can see here, this is the transcription factor name, and this is if you click on each of them, they will show you the matrix, the sequence logo. And you can see all of them, the TF is different. JunB, Jun, MAF, TBX, Bach1, and NFE2. And they are, this one is a little strange, but all the rest are in the same leucine zipper family of transcription factor. And if you click on each of them, their motif look quite similar. But you can use ChIP-seq to look at transcription factor interactions. For example, supposedly this transcription factor 1 is interacting with another transcription factor 2. And this interaction can happen, the 1 and 2 are binding right next to each other within the same, module. Remember, cluster of binding that can happen within the same 150 to 200 base pair region, or it could be a big loop, like it binds to 1, and then there's like some other loop on the DNA, and then the transcription factor 2 is binding far away, but because they interact, in the cross-linking step, you will cross-link the TF1 to its DNA, TF2 to its DNA, and also you will cross-link the TF1 and TF2 together. So it will form a chunk together. And then when you sonicate, this will be sonicated into shorter pieces. And so when you sequence the data out, you might notice, for example, in this case, within this peak, it has the motif of TF1. It also has the motif for TF2. But in this case, one peak only has, because you use this antibody to drag down, this piece of DNA will be enriched, but this piece of DNA will also be enriched because they're all together. And so you might see that if you do ChIP-seq of TF1, there are some regions that only have TF1 motif, but there are also regions that only have TF2 motif. And that is an indication that these two transcription factors work together. Okay, so you can look at this from two ways. The one is on the system, there is something called toolkit. If you just enter the ChIP-seq data of one transcription factor, it will automatically tell you what other transcription factor ChIP-seq has a peak that overlaps. So you can imagine if TF1 and TF2 collaborate or they interact a lot, whether you do TF1 ChIP-seq or whether you do TF2 ChIP-seq, their peaks will have a lot of overlaps, right? So, so the system database have the system toolkit. You enter some ChIP-seq data, it will pull out all the other ChIP-seq data that have significant overlap with your data of interest. And that can help you find out whether something overlap. For example, if we do nanog ChIP-seq, at first we'll say, there are other nanog ChIP-seq that look really like this. But also you will say, SOX2 also has significant overlap with your factor because SOX2 and nanog, they interact a lot as transcription factors. Okay, so you can look at these type of interactions based on the peak overlap. You can also look at this directly based on the motif enrichment. So for example, if we were to do this is actually a nanog ChIP-seq, you will see that the nanog motif is here, but you also see the SOX motif in here that are enriched. Right? So you can see it's a SOX1, sorry, SOX4, SOX11, and SOX10. They are all actually SOX motifs because the same family member. Right? So you know that the nanog family of transcription factors and the SOX transcription factor, they interact with each other. Okay, yes, Richard.
Sorry, I think I missed something. For the first case, how do you see an overlap when they're distant on?
Oh, those two sequences can be far apart, as far as a few kb or 10 kb or 100 kb probably even. It does happen. Yeah, so sometimes when you do a ChIP-seq, you will see that there is a motif. Your TF's own motif does not appear in some sequence. Instead, another transcription factor motif is significantly enriched in your data. That's an indication. Well, there's also a possibility that you use the wrong antibody, sample swap, right? But hopefully you find, yes, my factor motif is enriched, but then there is another motif that is also enriched, usually a secondary motif. And that's an indication that these two factors interact with each other. But remember, so here you can see if we do the nanog ChIP-seq, yes, in the very top we see the nanog motif, but then you see the SOX motif. You see SOX4, SOX11, SOX10, but there's a SOX2, SOX3, right? Which one is the one? Very often it's the one, is the SOX that's the highest expressed. That's interact in this cell type, that's interacting with your nanog of interest. Okay. Yeah, so either by a peak overlap or motif enrichment, you can figure out whether some family of transcription factor can interact with each other. Yeah, and then to really figure out which member is the specific member in the family is interacting, usually you look at expression. Yeah, so first of all, this transcription factor needs to be expressed fairly highly in the cell you are interested in. And so do I have an example? Oh, we do have an example. We do have some time. So in the very early days, this is a study that we conducted with our closest collaborator, Miles Brown's lab, where they are interested in estrogen receptor binding in breast cancer. So majority of female with breast cancer have an overactivated transcription factor, estrogen receptor. And you want to ask, where does it bind in the genome? What do they do? And so this is actually in early ChIP days, we got 3,000 peaks in the genome, and reviewers were like, gosh, 3,000, you know, now it's like easily 10,000. At that time, we were able to validate, yes, those binding sites, you can experimentally validate there's binding enrichment. And then we submit that paper initially to Nature. And then Nature just said, oh, you just show that the transcription factor binds to the genome in those locations. There's no insight, right? Is there anything interesting that you can find? So our collaborator came to us to say, what else can you find? So we started looking for motifs, and we said, you know, in addition to estrogen receptor motif, there's another factor, which is in a FORCAD, is a family of transcription factors with a different motif. And so, but then which FORCAD is the relevant one? So we look at the many breast cancer data together to ask, out of all the FORCAD family members, which one is highly expressed, and also the expression is correlated with estrogen receptor? Because it's like these friends have to hang out together to do something together, so that their expression are usually correlated. And once we do that, FOXA1 came on top. And then our collaborator later on was able to show that when you knock out FOXA1, estrogen receptor can no longer bind to their location anymore. And so the FOXA1 not only is an important factor, it's kind of an essential factor for estrogen receptor to bind. And then we were able to get it published in Cell. So you can see that's how informatics really helped the experimental work to generate a useful hypothesis. Okay, and so that's how you use it to find important interaction partners.